dominated by Gabriela Slak-Cohen. And Stephanie is going to talk about exploring exciton dynamics in DNA chromophore assemblies. And the floor is yours, Stephanie.
Thanks. All right, so my name is Steph Hart, and I'm a grad student in Gabriela Slak-Cohen's lab. And today I'll be talking about some of my work using two-dimensional electronic spectroscopy to explore exciton dynamics in DNA chromophore assemblies. But before I do that, I definitely want to take a minute to thank Thomas and Maxine for organizing this CFBS online series. During what's been kind of a strange year for scientific communication, this has been a really exciting forum to see some pretty cool work within the 2D field. And so with that, like many of these talks, I'll start by motivating my work within the context of natural light harvesting. And so we know that plants, along with algae and bacteria, harvest sunlight with relatively high quantum efficiency, and they use this energy for downstream metabolic processing. And they do this by utilizing pigment protein complexes. And these pigment protein complexes are biological chromophores, such as chlorophylls and carotenoids, that are scaffolded within a rigid protein network. And so plants and natural systems use this relatively elegant molecular machinery to capture and transport energy within the first femtoseconds to picoseconds after photoexcitation with nearly 90% quantum efficiency in a lot of cases. And so to aid in this relatively efficient energy transport process, these pigment protein complexes have very fine-tuned chemical interactions, and that includes the chemical interactions between chromophores themselves, which are responsible for generating delocalized excited states, as well as between chromophores and the surrounding environment, which are responsible for driving any kind of dissipative dynamics. And so because these natural light harvesting systems are so efficient, they've often been studied within the context of sort of model systems or generalizable principles for condensed phase energy transport, in hopes that at some point we could extract design principles that would have implications for artificial forms of light harvesting, as well as the design of molecular electronics.
Stephanie.
However, yeah.
Excuse me for the moment. Could you please get a little bit closer to the mic?
Oh, yeah, I'm so sorry about that. Please let me know if I'm too quiet again. Is this a little bit better?
That's a little bit better.
Okay, cool. Great. I definitely apologize that if I didn't repeat anything, please let me know. And so before we can take this kind of blueprint approach in studying light harvesting, we need a clear picture of what the explicit molecular parameters are that control light harvesting,
as well as how these parameters can be emulated and then controlled in an artificial system. And so today I'll be discussing some of my work in the Slak-Cohen lab. We've been using DNA chromophore assemblies to essentially emulate the pigment protein complexes found in biological systems. And so with this DNA platform approach, I'll be discussing how we can use this platform to control some of the molecular parameters that underlie light harvesting and exciton dynamics, as well as how these parameters play into more macroscopic performance quantities, such as the overall energy transport efficiency. And so to dive in a little bit more to these nanoscale light harvesters and their requisite sort of architectures, very much like plants use proteins as well as biological pigments to generate these pigment protein complexes. As a lab, we've been using DNA origami as a scaffolding structure to replace the protein, where here we can use sort of leverage the Watson and Crick-based pairing to generate any kind of arbitrary 1, 2, and 3-dimensional structure with a fair bit of spatial control over the systems that we're designing. And in conjunction with the use of synthetic dye chromophore systems, we can access a large portion of the visible range, which in total gives us a relatively tunable excitonic system with which we can start to kind of explore some of these questions as to what are these key parameters that underlie exciton dynamics. And so when we think about the interactions that control condensed phase dynamics, we typically lump them into kind of two key camps. The first being system parameters, and the second being environmental parameters. And when we zoom in on these DNA origami constructs, our system parameters
are comprised of electronically excited molecules, whereas our environmental parameters are comprised of the surrounding bath, so any kind of solvation or DNA environment. And so we're particularly interested in kind of the dynamic interplay between these two different sets of parameters, as there's our key kind of driving the resultant dynamics that we see in a lot of both natural and artificial light harvesting systems. And so first we'll kind of introduce the knobs that we're able to kind of control with this tunable excitonic system. In terms of system parameters, we're able to control the molecular structure, which drives the transition energy of these types of systems. And we're also able to control the intermolecular or electronic coupling between subsequent chromophores. And this is what allows us to generate delocalized excited states, very similar to what's seen in any kind of natural light harvesting system. And in terms of environmental parameters, we typically think about these in terms of fluctuations. And so short timescale fluctuations, which determine the short timescale dynamics, are typically driven by solvent interactions and occur on sort of the femtosecond to picosecond timescale. And long timescale fluctuations are typically determined by the structural scaffold environment, either the protein or in our case, the DNA. And these control dynamics on sort of the nanosecond to millisecond timescales. And so I'll start off by introducing some of the optical measurements we've done to sort of characterize our system parameters in this DNA origami structure. And so in kind of discussing the nanoscale assembly of these constructs, our chromophore of interest that we've been scaffolded into DNA is a cyanine dye, Cy3, which you can see here. And we use phosphoramidite chemistry to scaffold these directly into the DNA backbone to generate these structures. And so Cy3 exhibits a nice visible range absorption feature along with some vibronic transition and a relatively high quantum yield. And when we scaffold multiple cyanine chromophores into the DNA scaffold, such as generating a dimer or a trimer structure, we generate signatures of subradiant aggregation or H-type aggregation, which we can quantify through the emergence of a blue shifted band in our spectrum, as well as a decrease in the quantum yield. And so this gives us a handle for effectively saying that we have some kind of controlled extent of aggregation, kind of going from a monomer to a trimeric structure, and gives us kind of a base of going forward in hopes that at some point we can kind of work towards generating some of these very nice multi-excitonic or multi-chromophoric systems that are seen in natural light harvesting. And in a similar fashion, we can also explore the role of electronic coupling within a given dimeric system. And so if we look at the 2D electronic spectrum, we have this femtosecond correlation between the excitation and emission frequencies, as I'm sure a lot of people here are quite familiar with looking at. And in addition to diagonal features, we also have this upper diagonal crosspeak, which is a signature of electronic coupling. And if we build up these dimers to sort of look at a series of dimers that we can generate by separating each cyanine monomer constituent by a variable number of DNA bases, we can generate coupling from the relatively strong range to the relatively weak range in this type of system. And so here the electronic coupling is determined by the spatial position as well as the relative angle and orientation between the constituent chromophores as we go from the strong coupling range to the weak coupling range, and is mapped out relatively clearly with these changes in the upper diagonal cross peak as well, as they scale somewhat inversely with nucleotide spacing. And with this system, we also worked with some collaborators in Mark Batte's lab at MIT to do some molecular dynamic simulations of these types of dimers. And this allowed us to explore a heterogeneity in the types of sort of subpopulations that we can recover from these types of dimers that are formed and how they interact with their DNA environment. And so this allowed us to kind of track these different conformations as well as calculate what the expected electronic coupling would be from these different configurations recovered from our MD results, which track relatively clearly with our tracking of the upper diagonal cross peak from our 2D spectrum. And so this kind of in total shows us that we're able to generate a scalable system where we can control coupling from the relatively weak range on the order of tens of wave numbers to the relatively strong range on the order of hundreds of wave numbers to kind of access the regime that's typically seen in natural light harvesting systems. And so to kind of go back to our picture of sort of molecular knobs that we can tune in our system, we've checked the box on molecular structure as we can see by tuning our system to include variable extents of aggregation, as well as our control over intermolecular or electronic coupling, which allows us to essentially control the level of delocalization within our system. And so now I'll turn to our investigation of environmental parameters, starting with short time scale dynamics. And so we know that the 2D spectrum serves as this femtosecond timescale correlation plot between the excitation and emission frequencies. And due to this high resolution, we are allowed to essentially watch fluctuations in the transition energy as they occur on the excited state population, or on the excited state potential energy surface. And so at very early times, we have a strong correlation between excitation and emission frequencies. And owing to spectral diffusion at long time scales, we lose this correlation. And so here I'm going to borrow a tool that's more often used in the 2D-IR community, which is a centerline slope analysis to quantify this, this decorrelation as a function of time scale, where the centerline slope under a certain number of approximations is proportional to the frequency correlation functions. We can basically use the time scale of these dynamics to kind of uncover how the system is fluctuating on the femtosecond time scale. And so to explore these, the spectral diffusion in these different environments, we looked at cyanine dyes free in solution, as well as scaffold within a DNA environment. And when we look at the femtosecond timescale dynamics, we see a fast exponential decay within the first hundreds of femtoseconds after photoexcitation, which corresponds to spectral diffusion. And we also see these oscillatory features that are a function of basically vibrational modes coupling to the electronic transition, giving rise to the oscillatory features in this frequency correlation function. And when we, and these likely correspond to sort of a sum of low frequency modes that we can pull out from some transient gradient measurements. And so in comparing our sort of different environments of a free dye and a DNA duplex, we can see that the free dye experiences a slower overall sort of loss of correlation, as well as stronger coupling to these vibrational modes, which is likely due to a relatively fast decorrelation in the DNA environment, likely due to kind of the charged environment that it's residing in. And so to kind of clean up this picture of how these environmental effects dictate the femtosecond timescale dynamics, in addition to exploring our system in sort of a free dye environment, as well as in a relatively simple DNA duplex, we also looked at a monomeric species in these higher order double crossover tiles, or DX tiles, which are essentially two sets of duplexes bound together such that the persistence length is doubled, which generates a much more rigid environment. And these DX tiles are actually sort of the building blocks for generating higher order sort of 3D structures within the DNA origami community. And so when we look at the femtosecond timescale dynamics within these DX tiles, we see that they track more or less with the DNA duplex, highlighting in this case that these DNA-bound chromophores show similar behavior despite being in a somewhat different macroscopic environments, which is kind of interesting because here we show that these short timescale dynamics are controlled by the solvation rather than the DNA environment itself, which then prompted us to take a look at long timescale dynamics and how these effects result in exciton dynamics. And so to do this, we again looked at the same two types of DNA motifs, which are the monomeric species constructed within a duplex, as well as within a double crossover tile. And here I'll take a slight deviation from 2D and introduce a much simpler approach, which is our quantification of fluorescence and anisotropy to essentially put a time scale on these long time scale fluctuations that we see in the system. And so these are done by basically polarization controlled fluorescent excitation and emission experiments. And here we can track the timescales of the sort of loss of polarization for these two different species. And so in the duplex, we have a loss of correlation on sort of the 190 picosecond timescale, whereas it's much longer on the order of about 620 picoseconds in the case of the DX tile. And this kind of is pretty consistent with our sort of picture of understanding rigidity within these DNA structures, simply because the duplex should introduce a less rigid environment. And when we think about these fluctuations from the anisotropy within the context of a relatively simple microscopic model, we can also pull out some differences in sort of a fluctuating cone angle that allows us to kind of model our chromophore on top of an already fluctuating scaffold. And so with this, in addition to the differences of time scale, we also see a difference in these angles, thus suggesting that the nanosecond timescale or sort of long timescale fluctuation dynamics are indeed controlled by these different scaffold environments. And so this kind of checks our final box on how we can control these different system and environmental parameters over both short and long timescales. And so I'll spend the last few minutes of my discussion kind of on the second, highlighting a sort of motivating question that I brought up in my introduction, which is how do these impact energy transfer, or sort of more rigorously, any kind of functionality in an artificial condensed phase system. And so to do this, we took our structural DNA motifs of a duplex and a DX tile, and we added a lower energy psi5 or trap state, where we can basically track energy from the psi3 to the psi5 in a relatively clean fashion. And when we look at the steady state energy transfer efficiency, we somewhat surprisingly see that the duplex recovers a nearly 20% higher steady state energy transfer efficiency, despite the fact that these two systems are constructed such that the donor to acceptor distances are the same, and the monomers themselves exhibit nearly identical quantum yield spectral profiles and emissive lifetimes. And so this kind of interesting difference prompted us to go kind of back to the drawing board and think a little bit about what the orientation factors are looking like within these two different systems. And so kind of within this picture, we are considering most chiefly the transition dipole moment between the donor and acceptor in our system. And we can recover these distributions from molecular dynamics simulations, which basically shows these nice sort of bimodal distributions. And in addition to the general structure that these types of distributions play a role in, we also want to think about the time scales at which they fluctuate relative to energy transfer. And so in the limit of very fast conformational fluctuations, it's essentially only the mean that plays a role in determining sort of what the most important donor to acceptor angle is in this case. Whereas on the time scale of on the time scale of slow conformational dynamics, we kind of approach the limit of static averaging, which essentially means that this general structure, or in our case, sort of the bimodal structure of this distribution, comes to play into a role, or sort of a larger role, in the energy transfer process. And so mapping this back onto our time scales of depolarization, the duplex with its relatively fast depolarization on the 0.19 nanosecond timescale likely corresponds to the dynamic limit of energy transfer, where you have this relatively fast sampling of transition dipole moments kind of before the energy transfer process occurs, whereas the longer timescale depolarization of the DX tile likely corresponds to the static limit, where we have these much slower conformational configurations, and the general structure of these dipole angles likely plays more of a role in the energy transfer efficiency that we recover. And so to test this, we used a Monte Carlo fret simulation that incorporated these dynamic and static limits along with their relative distribution and dipole angles, and we're able to recover a relatively similar trend to what we recovered experimentally, which shows that, somewhat interestingly, in the limit of a relatively flexible scaffold, which is what we see in the duplex, we actually have higher energy transfer efficiency than on the relatively more rigid DX tile, which is somewhat non-intuitive, but it's kind of interesting because, one, it shows us that we have this kind of scaffold handle on how we can determine energy transfer efficiency, but two, is it somewhat non-intuitive because we typically think about energy transfer being efficient in these sort of rigid environments, but in this case, our ability, or sort of the DNA's ability to kind of sample around a wide range of different configuration actually allows it to recover a faster or sort of more efficient energy transfer process, which kind of highlights the role of the scaffold in sort of tuning these sort of large scale dynamics. And so to wrap up what I've been discussing today, we talked a little bit about how in these controllable excitonic systems, we've demonstrated control over both the molecular structure as well as intermolecular coupling through formation of some relatively well-controlled molecular aggregates, as well as control over the extent of delocalization within a dimeric structure. And in terms of environmental parameters, we use optical methods to characterize both the short and the long time scale dynamics that we can see in these systems. And in terms of bringing these all back to functionality, we showed that the long time scale dynamics dictated by DNA scaffold itself are going to be the most kind of key parameter in determining the relevant energy transfer sort of regimes that we're able to recover. And somewhat interestingly, we uncovered a situation where a more dynamic system in the case of a duplex actually transfers energy more efficiently, which could be kind of a design principle for building up higher order DNA structures moving forward as you kind of build towards this picture of sort of artificial light harvesting and generating these sort of artificial reaction centers and light harvesting complexes. And so with that, I definitely want to take a minute to thank the Schwab-Cohen Lab, which are a whole bunch of awesome scientists. So thank you. So my Professor Gabriella Schwab-Cohen, as well as another student in the Schwab-Cohen Lab, Weizhou Chen, and some of our collaborators at the University of Bern, as well as the MIT Bioengineering and Chemistry Department, as well as funding sources from the NSF and the DOE. And so with that, I'd be happy to take any questions.
Thank you very much. Questions indeed. So if nobody else have a question, maybe I can ask one. I think these DNA bounded dimers, they are very nice systems, right? So you can essentially look at the couplings between the dye molecules. Did you compare this with like the standard type of coupling models, like the dipole-dipole coupling or extended dipole or something like that? And to what extent does these coupling models then give the right answer?
Yes, that's a good question. We did think about these in the terms of just sort of the traditional sort of dipole-dipole coupling. In our case, because there is some displacement along DNA, the actual distance between base pairs is slightly different. And so that's kind of what was sort of the most key difference in these cases in terms of recovering differences in sort of a transition dipole approach. But they more or less showed the same trend. In our case, the kind of the useful part of the MD simulations was one, using a transition SDQ approach for calculating the electronic coupling, but two, it allowed us to look at sort of these different populations. So yes, it more or less scaled, but we did see just some definite differences in using a more rigorous MD-based model.
Okay, Vivek, please go ahead.
Hi, I had a quick question just so that I'm understanding the summary. So about the role of the environment, I guess to summarize, would it be correct to say that the short time fluctuations are controlled by the not by the scaffold, but the I guess the solvation environment, not the ions on the electrostatic interactions from the DNA backbone, and then the long-term fluctuations are the one controlled by the backbone. Is that the right way to say it?
Yeah, that's what our results have kind of indicated. There could definitely be some more work in the space, particularly in looking at the solvent dependence of particularly the short time scales. So we just considered this in the case of a sort of free dye in solution, as well as here, I can go back to these slides actually, probably helpful. We've looked at a free dye in solution as well as within this sort of DNA scaffold itself. And it's actually kind of weird that we saw this faster dephasing within a more rigid structure, but this is probably because when we are building these things, they're essentially replacing a base within the DNA backbone. So they're exposed to this very highly charged environment, which likely drives correlation. But yes, it's kind of the crux of it is the very fast time scales are driven by sort of the in DNA versus not in DNA environment. And then the long time scale dynamics are driven by the local fluctuation of sort of a nanosecond time scale within these different DNA motifs.
Okay, so I guess the faster fluctuations are determined by the electrostatic environment because of the DNA backbone. Is that what you're saying?
Yeah, that's that. Yes, exactly.
Okay. All right. Thank you.
Okay, more questions?
Maybe if I'm allowed to ask one at this slide, actually, because you see this intra molecular vibrational mode, which is clearly at 500 wave numbers. Can you say something more about the nature of this mode?
So we did some work looking at some more rigorous work looking at some theory results for just the Psi3 not in DNA on the system, and we actually recover modes that are along the order of around 400, around 550, and 600 wave numbers. And so we're thinking that this probably corresponds to some kind of superposition of these modes. And in our results of just like sort of like DFT Gaussian sort of B3 lit model, it corresponds to something relatively delocalized over the entire backbone. So it's sort of heterocycle sort of motion coupled to this sort of like polyene stretch. So not very descriptive in this case of sort of a very highly delocalized stretching motion. But we're thinking that it kind of probably corresponds to some kind of superposition of all of these low frequency modes.
Okay, thank you.
Can I ask a question? Stephanie, when you go to the anisotropy, could you please go to that slide?
The what slide?
The anisotropy slide.
Oh, yeah.
Yeah, so here, yes, right. So here well, you have here a monomer and in duplex monomer, right? So can you tell something about their dipole moments in these two systems? What I mean to say is that can you describe the system with a single dipole moment of a certain orientation or for instance, the duplex monomer, you need two different two dipole moments that might be oriented differently.
So in this case, we're only having a single sort of monomer side 3. And so the dipole orientation, or the transition dipole, is oriented along, here I can pull up my screen pointer. It's along the backbone of the assigning chromophore. So in this case, there's really only sort of like one that's relevant. But when we're thinking about these fluctuations, it's sort of relevant to the backbone of the DNA. And so that's kind of what we were quantifying in terms of the next slide of sort of picking out these dipole angles.
Okay, thank you. Okay, more questions? I guess not.
Okay. Then we should thank Stephanie again, I think. And